Dear Chairman, dear guests, I would like to introduce the dermatological research group of the Hungarian Academy of Sciences that actually operates within the walls of the Department of Dermatology and Allergology at the Faculty of Medicine, University of Szeged. The research group was founded by Professor Attila Dobozi, and its current leader is Professor Lajos Kemény. We have several research topics that we are interested in, and we are carrying out experimental dermatology research in these topics. We are focusing to make two multifactorial skin diseases. One is the psoriasis, and the other is the acne. Within the research on acne, we are also focusing on the analysis of innate immune functions of the human skin, and we are focusing especially on the interaction of the pathogenic bacteria, Propionibacterium acnes, and the human keratinocytes. And actually, the topic that Benjámin Papp will talk about is very much connected to this topic, but he will talk about a newly coming area of our research, the adaptive immune responses induced by yet another human pathogen, the Candida albicans. We are also interested in the investigation of several monogenic skin diseases, so-called genodermatosis. This work is also having diagnostic aspects within the Department of Dermatology and Allergology. And we also have a long-running tradition in investigations on the biological effects of light. And I would like to show you some examples for the the results of our experiments. But before that, I would like to introduce you some facilities that we have in the research laboratory. We have a very well-equipped laboratory for cellular biology experiments. We are mainly working with human cells. We don't use animal models. It's because we are in a very close connection with our plastic surgery department, and this provides us a very good source for human cells. We have a well-equipped laboratory for molecular biology studies, in which we mainly performing RNA and protein expression studies. And yet we have a well-equipped flow cytometry laboratory with a fluorescence imaging system that makes us able to study very detailed features of cellular events and tissue morphology. So the research topics, the major research topics, we are mainly interested in the molecular biology aberrances in the non-involved skin area of psoriatic patients. And recently we carried out a cDNA microarray experiment to identify differentially regulated genes and gene networks that are operating aberrantly in the psoriatic non-involved epidermis, and we were able to identify two such networks. And right now we are engaged in the detailed characterization of these networks. As I already told you, the other focus of our research is acne, the pathogenesis of this very common skin disease, mainly affecting the adolescent generation. And we are focusing on the interaction of the pathogenic bacteria, the Propionibacterium acnes, with the human keratinocytes. And recently we have got to the point to identify that P. acne strains having different pathogenicity have different capacity to produce propionic acid, and it seems to be a pathogenic feature in the pathogenesis of the acne disease. And we could also show that P. acne strains that are faster growing are more pathogenic. They release more propionic acid and causes the acidification of the environment and enhance keratinocyte cytotoxicity. As I have already told you, we are also having research and diagnostic work in different type of monogenic skin diseases, the so-called genodermatosis. And whenever we identify novel mutations in these monogenic skin diseases, we always try to characterize functionally these newly identified mutations, and we publish the results of the identification of the novel mutations and their functional characterization. And I has already mentioned to you that for many years the research group is also engaged in the characterization of the biological effects of UV light. And within this round of work, recently we identified and characterized a novel molecule in keratinocyte UV response, the human CoFan molecule, and we identified a regulatory network which is under the control of this molecule. And just recently we also started to use plasma irradiation in a wound healing model, and we try to find out whether this novel treatment would have any therapeutic options in wound healing. And as I have already mentioned to you, our St. Györgyi Albert student, Benjámin Papp, has recently joined the research group, and he's working on the characterization of adaptive immune responses upon the induction of the human pathogen Candida albicans. So with this, I would like to call Benjámin and give his talk. Thank you very much.
Dear audience, I would like to talk about our research in the topic of computational immunology. Candida albicans is a well-known member of the normal human flora and can be found on the skin and mucosal surface of the healthy individuals. This diploid fungus grows both as yeast and filamentous form. The hyphae form is pathogenic and may cause oral and genital infections in humans. The interaction between Candida albicans and the human innate immune system has already been investigated in detail. The innate immune system differentiates the pathogenic and the non-pathogenic form in a non-specific way, including non-protein substances such as fungal oligo and polysaccharides. The particular role of adaptive immune system is less well described. We carried out a computational analysis for the full proteome of Candida albicans. By the methods of machine learning, using an already known algorithm, we carried out epitope prediction for the full proteome. On the prediction results, we based analysis of antigenic proteins, assessment of protein domain antigenicity, and analysis of age allele frequency and epitope sequences. To study the possible link between Candida albicans and the human adaptive immune system, first, let's take a look on the adaptive immune response. Antigen-presenting cells ingest pathogens and digest their proteins into epitopes. The antigen-presenting cell then expresses a molecule called MHC2, which binds the epitope and represents it to the T cells in the lymph nodes. This representation then induces the immune response. First, the number of non-allele-specific epitopes occurring in each Candida albicans proteins were identified. By gene ontology term enrichment analysis, we are able to see the functions of the most antigenic proteins. This analysis showed that most of the terms are related to cell membrane, cell surface, and transmembrane transport. Terms related to pathogenic hyphen morphology are proteins linked with filamentous growth. We can conclude that the recognition of the virulent morphology as beneficial and the most antigenic proteins can be found on the most easily accessible places for the human adaptive immune system. Next, we wanted to know whether the epitopes are located randomly within the protein sequence or they can be found on domains. Our study showed that about 72% of the epitopes are located within protein domains. By gene ontology term enrichment analysis, we are able to assess the functions of the most antigenic protein domains. This analysis showed that these domains are related to cation transport, transmembrane transport, oxidation and reduction processes, and the glycophosphatidylenosyl inositol synthesis. Recognition or blocking these transporters can be an effective strategy for the adaptive immune system to neutralize the pathogenic fungus. Oxidation and reduction processes play a significant role in the defense mechanism against innate immunity. The glycophosphatidylenosyl inositol synthesis is responsible for cell wall remodeling in case of yeast form turning into pathogenic hyphae form. Next, we examine the HLA allele specialization for the epitopes. For this, we use the common and well-documented alleles database. First, we compared the number of Candida albicans epitope recognized by common and uncommon alleles. Surprisingly, we found that common alleles recognize a lower number of epitopes than uncommon ones. By gene ontology term enrichment analysis, we found that common alleles, by a lower number of epitopes, recognize more functional groups than uncommon ones, and we found no functional groups recognized only by uncommon alleles. By the identification of only common HLA allele-related functional groups, we found they were mostly specific ABC transporters. These transporters play a huge role in pathogenicity, so attacking them is a good strategy for the adaptive immune system against Candida albicans. Although common alleles recognize a lower number of epitopes, we found this recognition cover more functional groups, and this specialization could mean an evolutional benefit for common HLA alleles. Finally, we were curious about the occurrence of Candida albicans epitopes in other species. For this, we used BLAST, which can find region similarities between nucleotide sequences. Our results are shown on a phylogenetic tree. Red color code shows the high occurrence of Candida albicans epitope, and green color code shows the low occurrence. As we expected, the highest similarities were found with other Candida species. Surprisingly, two non-fungal genera has also been found with high similarities: the animal parasite Aemera genus and the Drosophila genus. We found that Candida glabrata carry only a few epitopes of Candida albicans. This phenomenon could explain the whole different way of disease pathomechanism caused by the two relative species. It has already been published that Candida glabrata infection is associated with a lower incidence of inflammation and cellular infiltrate, which could be a coincidence of the low antigenicity of this fungus. Our conclusions. With our results, we've proven the essential role of adaptive immunity in case of candidiasis. Although our studies are computational, published experimental data validate our results. And our analysis pipeline can be suitable for discovering further interactions in the microbiome. Thank you for your attention.